One of the most important things that has to happen in the nervous system for it to be successful is the process of myelination. Myelin is a very important fatty protein that affects the ability of neurons to conduct nerve impulses. And those nerve impulses are called action potentials. They're so fast that they travel as fast as in the speed of milliseconds. Myelination is the process of wrapping the axon with a myelin sheath, kind of like you would wrap a cable with electrical tape. And the purpose of this is to electrically insulate it so that the signal travels down an axon much faster. And this happens in both the central nervous system and in the peripheral nervous system, in the glial cells that you just learned about. In the peripheral nervous system, there are neurolemmocytes, which are also called Schwann cells, which wrap the axon so that the signal travels much faster. In the central nervous system, the glial cell responsible for this is called an oligodendrocyte. And in order for this to occur, Schwann cells have to myelinate the axon, and it takes multiple of these Schwann cells. There's an outer membrane of the Schwann cell called a neurolemma, which is going to surround the axon. And in the central nervous system, a single oligodendrocyte can myelinate short portions of the axon. So we have multiple Schwann cells in the peripheral nervous system, but only one oligodendrocyte in the central nervous system. This micrograph is showing you the types of conduction that actually occur here. So what I want you to notice where we have this little green line, these are what are called nodes of Ranvier. They're also called neurofibril nodes or nodes of Ranvier. And this is important because this is where the action potential actually leaps to. So the goal of axons is to spread the action potential as quickly as possible throughout the nervous system. So this micrograph is showing axons that function in what's called saltatory conduction. And the word saltatory means to leap. So imagine that the action potential is literally leaping from one axon or along the axon to the next neurofibril node or node of Ranvier. So saltatory conduction, you should know that it is the fastest conduction, and it occurs because of this myelination. So within the peripheral nervous system, it's the Schwann cells or the neurolemmocytes which are forming the myelin sheath. So notice here first that we have the main parts of the neuron, which would be the dendrites. Remember, the dendrites are responsible for taking the incoming information to the neuron cell body, also called the soma. And the axon takes the information away from the cell body. And because of this myelin, it leaps from node to node. And those nodes are called neurofibril nodes or nodes of Ranvier. And this sort of leaping that occurs is extremely fast, and it's called saltatory conduction. So when we don't have this myelination that occurs, the signal is going to travel much more slowly down the axon. So in this slide, we can see a myelinated axon, and the myelin is shown here within this bracket region where I have the laser pointer on the slide. So notice that is the myelin sheath. This is pointing out an unmyelinated axon. And in unmyelinated axons, we have a different type of nerve conduction, which is called continuous conduction. This means that the nerve impulse is going to travel the entire length of the axon at a very slow rate and much slower than saltatory conduction. So what you need to know is that there's two types of conduction that occur. There's the impulse conduction that occurs in a myelinated axon called saltatory conduction, where the impulse jumps or leaps from one neurofibril node to another. And it happens in large myelinated axons, such as those that conduct impulses to skeletal muscles in the limbs. And on the micrograph on the right side of your slide, this is showing a myelinated axon. Contrast that with an unmyelinated axon. We don't see any myelin that's surrounding that axon. And thus the signal is going to travel at a much, much slower rate. Nerves are cable-like bundles of parallel axons, and you should know the connective tissue membranes that surround nerves. And the nice thing about this is that the prefix or the terminology is very similar to that for muscles that you learned. So surrounding one individual axon, first of all, is the myelin sheath that we just learned about. So the connective tissue layer around it would be the endoneurium, and the endoneurium is surrounding each individual axon. Within this region is a group of axons which organize into a fascicle. A fascicle is a bundle of axons. Recall that you had the term fascicle in muscles, and surrounding that fascicle is a perineurium, which is the connective tissue layer around it. Then surrounding the entire nerve, which is a cable-like bundle of axons, is the epineurium. And the epineurium surrounds the entire nerve. So, for example, the longest nerve in our body happens to be called the sciatic nerve, and it's surrounded by a connective tissue membrane called the epineurium.